Hello, everybody, and we're going to get started with DNA replication part 1. So, structure and function of the double helix, of course, very tightly related. The function of the double helix is to carry our genetic information, so it needs to be pretty simple to copy, simple to explain, and basically the rules of base pairing is kind of the basic tenet behind how it's so easy, how and why it's so easy to copy DNA. Okay. Since the two strands are complementary, each strand can act as a template for building a new strand. Okay. So if we have our parent molecule here, we separate the strands, and then each strand can act as a template to make a new strand. Okay? Because the rules of base pairing are definite, you get two identical copies of the same molecule. This is called semiconservative model of replication. So each daughter molecule has one old strand, that's the conserved part, and one new strand. DNA is remarkable in its speed. We'll talk about it again later, but in eukaryotes, 50 nucleotides per second, and in prokaryotes, 500 nucleotides per second. And also accuracy. It's got to be copied correctly. Okay, lots and lots of enzymes are involved in making DNA happen, replication happen. Okay, so DNA starts, we have to start somewhere at an origin of replication. So the DNA strands are separated, and the resulting structure is called a replication bubble. Okay, eukaryotic chromosomes, because they are much longer and much bigger, have hundreds or even thousands of origins of replication to make it go faster. And replication proceeds in both directions from the origin until the molecule is copied. So it looks kind of like this. So in prokaryotes over here, there's just one origin, and the bubble opens up. This is a replication bubble. And each bubble has two replication forks, one on each side. And you see replication proceeds away from the origin, okay, and eventually comes around and until the ends meet, and you're left with two identical molecules. Works the same way in eukaryotes, except the chromosomes are linear, and there are multiple origins of replication, so you get multiple replication bubbles. Okay, replication moves away from the bubble in the direction of the fork. Okay, and then when the forks meet up, bam, you got two DNA molecules. And here's some micrographs of, like, here's a bubble, and these are pointing to forks. Same thing. It's a little easier to see in the prokaryote. There's your replication forks, and here's your replication bubble. We talked about the fork on the previous slide. And then the separating of the strands at the origin is done by an enzyme called helicase, and this is kind of the first of the proteins that are involved in DNA replication. So helicase separates the strands, and then the job of the strands is to be together. So if you don't do anything after you separate the strands, they're basically going to snap right back together. So there are proteins called single-stranded binding proteins, or SSBs, and they prevent the strands from coming back together. Okay, ahead of the fork, when you're kind of untwisting one end, and before the fork, it gets very, very tightly twisted. And topoisomerase is another enzyme, ASE, that fixes this overwinding problem of DNA ahead of the fork, and this keeps the DNA from getting super twisted up and breaking. So this kind of shows you, puts it all together. Here is helicase. It is unzipping so this is my replication fork. Okay, right here with the strands split apart. Helicase is unzipping or separating the strands. Here's some single-stranded binding protein. that are keeping the strands from separating. And then here's topoisomerase working ahead of the fork in order to prevent overwinding. Okay. We're going to talk about primase in a second. Actually, now. The main job of replication is done by a DNA polymerase, an enzyme that makes a polymer of DNA. But they're limited in that they can't initiate synthesis. They always have to add nucleotides to an existing 3' end. So because they can't initiate synthesis, the initial nucleotide strand is a short RNA primer that's laid down by this enzyme primase. So primase is an RNA polymerase. Okay, and it can start a strand. It can kind of put that first base down and then add another base. Primers are about 10 nucleotides long. And then once it puts down this little RNA primer, primase will leave off and will pick up with DNA polymerase. And, this basically explains what I just said. So primase is a RNA polymerase, starts a little strand of RNA molecules, and that can use 5 to 10 nucleotides long, and now it's got a free 3' end. That will be the starting point for the new DNA strand. Okay, and we're going to pick up with the rest in part 2. So go on to, that is not one word, on to part 2. All right, go get him, Tiger.